My name is Emma Dirt, and this is my annotation of the Waxworm genome. So, during our lifetime, there has been a significant increase in plastic waste, causing environmental pressure around the world. In NC State's Genetics 312 lab, we focused on annotating the genome of a larval moth that has the ability to quickly break down plastic with its saliva. We used technology and biological data to annotate the genome to find the specific genes that allowed the waxworm to quickly degrade plastic. This information is crucial for future research as it can help address environmental issues. As stated, plastic pollution is one of the most pressing environmental challenges of our time. Over 300 million tons of plastic is produced annually, with nearly half of that designed to be single-use. Unlike natural materials, plastic can take centuries to decompose, causing it to accumulate in landfills, oceans, and throughout our ecosystems. Plastic pollution is prevalent worldwide and does not seem to be slowing down. So what can we do to help? In this class, we have been researching a species called Galeria melanella, commonly known as the waxworm, which is a common pest in beehives. Waxworms span 4 life stages: egg, larvae, pupae, and adult. First, eggs hatch in the larvae, which is the actual waxworm stage. These small caterpillars will feed on beeswax, pollen, and honey from anywhere between 1 to 5 months, where they grow up to 2 centimeters in length. At this stage, they have a special ability where they can break down plastics with their saliva. After 5 months of feeding, the larvae will spin themselves into a silk cocoon where they will pupate for 1 to 2 weeks before emerging as an adult moth. The adults only live for 7 to 10 days where they focus on mating and laying new eggs. Once eggs are laid, they will hatch within 5 to 8 days. This rapid life cycle makes the waxworm a significant pest for beekeepers. Despite this, the worms are able to address a serious environmental challenge. On the right, I have a life cycle picture of the Galeria melanella waxworm, where you can see we go from egg all the way to cocoon and moth. And then on the bottom left, I have a picture going from eggs to, again, cocoon, pupae, and moth. So, research has shown that waxworm larvae have the ability to break down polyethylene plastic due to a special component in their saliva. The key enzymes are identified as Demetra and PEase, which work by oxidizing and breaking down the chemical structure of plastic at a rapid rate. This was first documented in 2017 when scientists observed waxworms creating holes in plastic bags. It is theorized that the waxworm has this ability due to an evolutionary response to digesting beeswax, which is their natural food source. Beeswax shares a similar chemical structure to polyethylene, which helps explain why waxworms can dissolve it so easily. Unlike traditional chemicals used to break down plastic, these enzymes work at room temperature and have no additional stipulations, making them promising for environmental applications. Current research on these enzymes is focused on isolating and enhancing them for large-scale plastic waste management. On the left, I have an image of a waxworm eating through a blue plastic bag. So, we are able to find and isolate the genetic sequences related to this enzyme by a process called genome annotation. This is the process of identifying and labeling specific elements within a sequence of DNA, which is similar to marking a landmark on a map. Scientists use computational tools and biological evidence to find specific sections of a gene, and these sections are made of a small nucleotide, which are letters that code for a protein. Genome annotation takes these letters and puts them into a readable sequence of code, which acts as an instruction manual for how that organism operates. For the waxworm, genome annotation is important because it allows us to pinpoint the exact genes that are responsible for producing the plastic digesting enzymes, such as the previously mentioned Demetra and PEase. With this information, scientists can research how to use this enzyme to engineer solutions for real-world problems, such as plastic pollution. In summary, annotation turns a string of DNA into readable information that we can use. On the right, I have an image of the central dogma of molecular biology. This concept is the principle of genetics, and it shows the process of going from DNA to RNA to protein, having transcription, splicing, and translation in between those steps. Throughout this class, we learned how to use a program called Galaxy. This program allowed us to annotate and read the genome sequence of the waxworm, which increased our understanding of the plastic degrading enzyme. To start this process, we first uploaded the waxworm's raw genetic data and added RNA clues to help us find the sections related to the enzymes. We then used a tool called FASTA statistics to make sure that there were no missing sections from the DNA. We then marked the repetitive sections to avoid confusion in our code. Next, we used the Maker tool to combine specific data and compared it to the genome of a fruit fly. This comparison gives us an idea of where the genes start and stop in the specific genome. This allows us to segregate them and focus on the characteristics of the plastic degrading enzyme. Next, we use Busco, which is a tool that looks for must-have genes that the waxworm has to have to survive. These genes help assure that our annotation was done accurately. Lastly, we used JBrowse, a tool that allowed us to explore a map of our annotated genes. This let us zoom in and out of areas to see specific codes. For future steps in this project, I would like to suggest looking into different tools on Galaxy that could speed up the process or make it a little bit less stressful on students, as some annotation steps took a lot longer than others. In addition to this, I think that publishing our annotation histories and sharing them with future students could help the longevity of this process, as science is all about trial and error. Thank you!